I want you to imagine, just for a second, trying to wire a supercomputer from scratch. But the catch is you are in total darkness.
Right. Total darkness.
Yeah.
And no instructions at all.
Exactly. You have zero blueprints. And your only tools to build this thing are, a series of fleeting chemical signals.
It sounds like an impossible engineering problem, honestly.
It really does. But, that is the literal reality of what a developing brain has to do every single time it forms.
Yeah, millions of connections and just massive cellular complexity. And they are organizing themselves in the dark as we speak. We're only just beginning to truly decode the biological algorithm that makes it happen without the whole system just, crashing.
Which brings us to today. Welcome to this deep dive. Today, for you listening, we have a very specific mission. We are going to examine this really dense, fascinating 2026 paper from Nature Neuroscience.
It is a phenomenal paper.
It is. And our goal is to dissect exactly how a brain generates dozens of highly specific specialized cells from what looks like a totally uniform starting point. And to do this, we are using the Drosophila as our Rosetta Stone.
Right, to the fruit fly.
Yeah, specifically, we're looking at its optic lobe. So let's just set the stage here.
Sure, so the Drosophila Optic Lobe is this structurally beautiful vision center. It's made up of the lamina, the medulla, the lobula, and the lobular plate.
Right.
But our primary focus today, and really the main focus of the paper, is this tiny cellular nursery.
The outer proliferation center. Right?
Exactly, the main outer proliferation center, or OPC. This is the origin point where all the medulla neurons are literally born. And it serves as a perfect model for us because the mechanisms we find in the fruit fly, well, they often represent these universal biological truths that apply to human brains too.
I just find the mechanics of that microscopic universe incredibly compelling. So let's trace the actual biological algorithm that builds the structure. Because before we look at how brain cells get their unique identities, we kind of need to map out the literal assembly line that produces them.
Right, the who and the how.
Yeah, and the foundational tissue here is the neuroepithelium, right?
Yeah, the neuroepithelium. Those are the raw building blocks. So from this neuroepithelium, you get this wave of development that converts these foundational cells into what we call neuroblasts.
Okay, neuroblasts.
And neuroblasts operate as neural stem cells. Their defining characteristic, the thing that makes them special, is that they undergo asymmetric division to self-renew.
Meaning they don't split evenly?
Right. They continuously split, but they leave one exact copy of themselves behind to keep the biological factory running, while the other cell moves down the assembly line to become something else.
So if we map this to, like, a corporate factory for everyone listening, the neuroepithelium is the factory floor itself. And that floor eventually spawns a CEO.
The neuroblast.
The neuroblast.
Yeah.
And the CEO stays in the corner office making these high-level decisions, but they delegate the actual production to an intermediate progenitor cell, which the paper calls the ganglion mother cell, or GMC.
I like that analogy.
Yeah.
The GMC basically acts as our middle manager. Its entire existence is dedicated to a very specific, limited task. It divides exactly once.
Just once.
Just once. And that single division creates two final postmitotic neurons. These are the specialized floor workers. And because they're postmitotic, well, they will never divide again.
Their fate is sealed.
Exactly. Their fate as functional neurons is permanently sealed.
Okay, but I have to ask, it seems, highly inefficient to have this ganglion mother cell just divide once. Because biology usually optimizes for energy, right?
Yes, yeah.
So I would think the neuroblast CEO could just make the neurons directly and cut out the middleman entirely, unless this intermediate step is setting up some sort of controlled split that the neuroblast just can't do on its own.
You hit the nail on the head. That is the exact mathematical elegance of the whole system. The GMC middle manager is completely necessary for an exponential increase in cell diversity.
Okay, how so?
By adding this intermediate step, the system sets up a crucial binary split mechanism. We call it notch signaling. It creates two sister cells at the exact same time, in the exact same physical space, but forces them to immediately diverge in their identity.
Oh, wow.
Yeah, we will explore the actual mechanics of notch signaling in a moment, but without that GMC, you lose that vital branching point.
So it's basically an evolutionary workaround to double the complexity of the output, but without needing a vastly more complex stem cell.
Exactly.
Okay, so now that we have our raw neurons rolling off the assembly line, we hit the real core question here. How do they know what to become? Because a medulla needs dozens of different types of neurons to actually process vision.
Right. They can't all just be generic workers.
Yeah.
And according to the paper, They rely on a biological 3D GPS system. And this system is built on 3 specification modules.
Okay, the biological GPS. Let's break down those 3 modules.
The first of those modules is spatial patterning. This answers the where. The main OPC, it isn't just a uniform blob of tissue. It is physically divided into distinct zones. And these zones are defined by specific transcription factors.
And a transcription factor, just to clarify the mechanism for you listening, that is a protein that binds to specific DNA sequences. It basically acts like a genetic light switch, turning particular genes on or off.
Yeah, which controls the cell's function. So in the OPC, these spatial transcription factors set up these vast chemical territories. You have specific domains governed by factors like VSX, optics, and a signaling molecule called DTP.
So it's basically a chemical grid.
Exactly. And there are also dorsal and ventral domains defined by SPALT, DISCO, and Hedgehog, or HH. The researchers mapped this out by looking at how these protein gradients physically overlap in space.
And the paper actually found some new boundaries, right?
Yeah, what this paper discovered is that the divisions are even more granular than we thought. They mapped a tiny region they call the VVSX stripe, and also a highly specific region where DPA and optics overlap, which creates these highly specialized microenvironments.
So if we stick to our identity analogy, this spatial patterning is akin to the country or the state you were born in. It provides a foundational geographic identity.
That's a perfect way to look at it. The second module, which gets layered on top of that spatial geography, is temporal patterning. This answers the when.
So time.
Right. Because as those ceonoblasts age, they don't just remain stagnant, they operate on a biological clock. So they sequentially express a cascade of different temporal transcription factors over time.
Like a schedule.
Exactly. First, they express a gene called eighth, then they transition to OPA, then A, and so on.
But wait, because the neuroblasts are arranged in physical space, this temporal clock must leave some kind of physical trace as it ticks, right?
It does. It creates concentric rings of gene expression in the newborn neurons.
Oh, like tree rings.
Exactly like tree rings. As the neuroblast ages and changes its temporal transcription factor, the neurons it produces inherit that specific temporal tag. So you end up with a spatial pattern that looks just like the rings of a tree, with each ring reflecting the exact developmental age of the stem cells that produce them.
That is so cool. So layering that onto our analogy, if spatial patterning is the country you are born in, temporal patterning is the historical era or the decade you were born in.
Right.
So a neuron born in the VSX spatial domain during the, say, the 8th temporal window, it has a completely different foundational identity than a neuron born in the exact same domain, but later during the I window.
Yes, exactly.
Yeah.
And that brings us to the third specification module, which is notch status. This is a binary switch we touched on earlier with the ganglion mother cell.
Right, the middle manager split.
Yeah. So when the GMC divides into 2 sister neurons, they share the exact same spatial and temporal coordinates. They were born in the same place at the same time, but they physically interact. And one neuron receives an active notch signaling pathway.
Let me make sure I understand the mechanics of that signal for the listener. Notch isn't just an abstract concept, right? It's a literal physical receptor protein on the surface of the cell.
It is a physical structure, yeah.
And when it binds to a ligand on the sister cell, a piece of the notch protein actually snaps off, travels into the nucleus, and changes the gene expression.
That physical cleavage, that snapping off, is the trigger.
Yeah.
The cell where that happens becomes notch on. The sister cell, which provided the ligand but didn't get its own notch receptor activated, well, it becomes notch off.
Wow.
Yeah, it forces an immediate binary divergence in cell fate between 2 sisters that are otherwise entirely identical in space and time.
It's literally like being born the older or the younger sibling. It's a binary 0 or 1 situation that completely alters your trajectory.
Exactly.
Okay, this is a lot of dense material, so let me just pause and consolidate this map we just built before we move into how the cells actually remember this information. Just a quick recap for you listening.
Good idea.
So, bullet 1, neurogenesis is an assembly line. Neuroblast to GMC, which then splits into 2 neurons. Bullet 2, every single neuron's fate is determined by a 3-part system. We can use a mnemonic here. The space-time switch coordinate system, space for the spatial domain where you were born, time for the temporal window when you were born, and switch for your binary notch status.
And if you think about it, the permutations are mathematically staggering. I mean, intersecting multiple spatial domains with multiple temporal windows, and then multiplying all that by 2 for the notch switch.
It's huge.
It generates a grid capable of producing incredible neuronal diversity from a very simple set of overlapping rules.
But this brings up a massive logistical problem. The space-time switch coordinate system, it only exists during the developmental phase. I mean, those spatial gradients eventually dissipate. The temporal clocks stop ticking once the neuroblast finishes its job.
Right, they are temporary.
So how does an adult fruit fly neuron weeks or months later remember its identity when those initial stem cell signals have completely vanished?
That is the big question. And the answer is that the developmental signals hand off their instructions to what we call terminal selectors, or TSs.
Terminal selectors.
Right. If the space-time switch is the transient signal, the terminal selector is the permanent record. Terminal selectors are downstream transcription factors, and their sole purpose is to continuously maintain the neuron's specific functional features for the remainder of its life.
Features like what?
Like its physical shape, its connectivity, and specifically the type of neurotransmitter it uses to communicate.
Oh, I see. So if the space-time switch is the university degree, you spend a finite amount of time there, you get your coordinates, and then the experience is over. The terminal selector is the actual career you practice every single day for the rest of your life.
Exactly. Once the terminal selectors are initiated by those temporary developmental coordinates, they basically create autoregulatory feedback loops.
Meaning they keep themselves turned on.
Yeah, they continuously bind to their own DNA promoters to keep themselves active, which in turn keeps the necessary effector genes active forever. And the truly groundbreaking aspect of this 2026 paper is that the researchers mapped specific terminal selector candidates back to their exact developmental origins.
Wow, so if this mapping is as precise as the paper suggests, there must be specific genetic footprints we can see in the adult cell that trace directly back to, say, the notch binary switch.
Oh, absolutely. The Notch-On module is a perfect illustration of this. The data shows that a gene called apteris, or we just call it AP, correlates almost flawlessly with the notch-on status.
Really?
Yes, every single adult cell that had notch turned on during development uses the AP terminal selector to help maintain its adult identity. The spatial domain it originated from doesn't even matter. If it was notch-on, it expresses AP.
It acts as a universal biological law for those particular cells. Okay, so what about the temporal windows? Does the tree ring leave a permanent mark?
It does. The researchers found that the 8th gene, which defined the very first temporal window we discussed, it actually pivots to act as a terminal selector later on.
Oh, so it does double duty.
Exactly. It is continuously expressed exclusively in adult neurons that originally spawned during that specific 8th temporal window. So the transient clock literally leaves a permanent genetic signature.
Okay, I have to stop and ask about the methodology behind this, because proving that continuous link sounds, frankly, nearly impossible. I mean, you are trying to map the wiring of a dense adult brain back to a fleeting moment in embryonic development. How do you do that? The paper mentions single-cell RNA sequencing, or cRNA-seq, as their primary tool.
Right. Single-cell RNA sequencing is how they read the final output. The process involves isolating individual adult cells, lysing or breaking them open, and capturing their messenger RNA.
Right.
And by reverse transcribing that RNA into measurable DNA, researchers can see the exact genetic recipe. They can see exactly which genes are turned on and off in a single cell at a specific moment.
Okay, reading the adult RNA makes sense. I get that. But how do you definitively link that adult RNA profile back to a specific neuroblast that existed weeks ago?
To bridge that gap, they utilized a brilliant genetic tracking system. It relies on GAL4 drivers and memory cassettes.
Memory cassettes.
Yeah. They genetically engineered the fruit flies to contain temperature-sensitive memory cassettes.
I have to pause on temperature-sensitive memory cassettes. How does temperature actually alter a cell's memory?
Okay, so the flies are engineered with a specific recombinase enzyme. But this enzyme only becomes active at a higher temperature, specifically 29 degrees Celsius.
Oh, I see.
So when the researchers raised the temperature of the developing flies to 29 degrees at highly specific times, the heat activated the enzyme. And this enzyme physically snips out a stop sequence in the cell's DNA.
Wait, so by removing that stop sequence, it basically permanently turns on a reporter gene?
Correct. In this case, it turns on a green fluorescent protein, or GFP. And because the DNA itself is altered, that neuroblast and every single neuron it ever produces will permanently glow green.
Wow. They essentially spray-painted specific stem cells green while they were still on the developmental assembly line.
That's exactly what they did. And once the fly reaches adulthood, they extract the brain, and they use a machine called fluorescence-activated cell sorting, FACS for short, to physically separate out just the glowing green neurons.
And then they run the single-cell RNA sequencing on them.
Exactly. They know the adult genetic profile, and because of the green tag that was activated at a specific developmental time, they know exactly which spatial or temporal domain it came from.
That is incredible engineering, but they must be looking at thousands and thousands of cells. How do you process that sheer volume of data?
Well, to handle the data, they implemented random forest machine learning models.
Oh, AI. If I recall my data science basics, a random forest works by building hundreds of different decision trees, training them on a subset of data, and having them vote on the most likely outcome to make predictions.
You've got it. So the researchers trained the algorithm by feeding it the developmental origins and the terminal selector codes. They basically asked the AI, given these specific developmental coordinates, can you predict the adult neuron's features, like its gene expression profile or its physical morphology?
And could it?
The random forest models predicted the adult features with remarkable accuracy.
Okay, but I have to push back a little on the use of machine learning here. Machine learning is, notorious for finding complex correlations that don't actually reflect a biological reality. How do we know the AI isn't just finding coincidental patterns? Like, does this actually prove that these terminal selectors are causing the adult features?
That's a great point, and distinguishing between correlation and causation is critical here. You were completely right that the random forest algorithm itself only proves predictive correlation.
Right.
However, the study relies on a foundation of previous physical experiments. Scientists have previously performed knockout studies.
Where they delete the gene.
Exactly. Physically deleting these specific terminal selector genes in living flies. And when they do that, they observe that the neurons fail to develop their proper adult features.
Oh, okay. So the physical knockout experiments established the causation beforehand, and the AI algorithm just maps out exactly where and how those functional rules apply across the entire optic lobe.
Precisely. The machine learning confirms that the system is highly ordered and predictable on a macro scale, which validates all the physical experiments.
Okay, let's do another quick recap for the listener to consolidate the technical side of this before we look at the evolutionary implications.
Yep.
So bullet 1. Terminal selectors are the permanent genetic managers that take over from the transient space-time switch to maintain identity forever. Bullet 2. Scientists prove this link using a whole tech stack. They use temperature-activated memory cassettes to permanently tag the cells with a green glow, single-cell RNA sequencing to read their active genes, and random forest machine learning models to predict and map the outcomes.
Perfect summary. We have the coordinates and we have the lifetime managers. So now we can look at the practical functional traits these terminal selectors actually control.
And neurotransmitters would be the most obvious functional output, right? Because they are the chemical language the brain uses to communicate. Does the paper show that the chemical identity of a neuron is tied to these developmental coordinates?
The chemical identity is deeply hardwired to the coordinates. Let's look at VATHT, which is a key marker for cholinergic neurons.
Meaning neurons that use the neurotransmitter acetylcholine?
Right. The study found that VATH is reliably activated by the AP terminal selector.
Wait, and since AP is the terminal selector for almost all notch-on neurons, that means simply being the older sibling in the notch binary switch almost guarantees a neuron will use acetylcholine to communicate.
Yes. Furthermore, markers like VGLUT, which defines glutamatergic neurons, and VG, which defines GABAergic neurons, well, they're tied to highly specific temporal and notch-off combinations.
Can you give an example?
Sure. For instance, a different terminal selector called TJ activates glutamatergic identity, but it is restricted to very specific temporal windows. So the chemical language a neuron speaks is a direct, calculated readout of its space-time switch.
Which brings me to a big evolutionary question. Because the brain is so meticulously wired, if a random mutation occurs, say a spatial domain gets physically split, or a mutation causes the temporal clock to tick a little longer and creates a brand new temporal window, you would think the whole system would encounter a fatal error.
Like a computer crashing.
Right. How do new neuronal types evolve without breaking the biological matrix?
The resilience of the algorithm is perhaps the most fascinating insight of the entire paper. The system doesn't crash because of the rigid structure of the space-time switch itself. When a mutation creates a newly born neuron with altered coordinates, that neuron isn't just a blank slate.
What is it then?
It automatically inherits modules' predetermined features based on the coordinates it does have.
Oh, I see. It basically comes with a built-in starter kit. Even if it's in a new mutated spatial domain, it still has a notch status, so it might default to being cholinergic.
Exactly. It will express baseline terminal selectors based on its temporal or spatial lineage, whatever parts of the coordinate system it still recognizes. This built-in starter kit biases these novel neurons to integrate smoothly into existing neural circuits.
That's brilliant.
It prevents catastrophic failure and allows evolution to safely tinker with new cell types, gradually adding complexity to the brain over millions of years.
So evolution isn't just throwing random parts into the system and hoping they connect. It is providing every new cell with a functional default resume on day one so it can integrate and get to work immediately.
It really shifts our whole understanding of neural development. It goes from a process of, mysterious organic growth to one of precise, predictable, and remarkably robust computation.
We have covered an immense amount of biological terrain today, and what I'm taking away is that the mind is not a random soup of cells. It relies on this three-dimensional coordinate system of space, time, and binary switches, which then hands off its instructions to permanent genetic managers called terminal selectors.
And understanding this exact pathway in the Drosophila Optic Lobe, it provides a fundamental blueprint for how complex nervous systems assemble themselves from seemingly simple starting conditions.
Which leaves us with a final thought to ponder for everyone listening. If a tiny fruit fly utilizes this incredibly precise space-time switch algorithm to build its brain, it is highly probable that our own human brains rely on a variation of this exact same code.
Almost certainly.
So imagine the possibilities in the field of regenerative medicine when we learn how to not just read, but actively write our own spatial and temporal coordinates into human stem cells. Could we one day custom print exact functional solutions to neurodegenerative diseases just because we finally have the source code?
It's an exciting future.
It really is. Keep questioning the world around you, keep exploring the science, and we'll catch you on the next deep dive.